Hey everyone, and welcome to this explainer. Today we are putting on our virtual lab coats and taking a massive deep dive into the seriously rigorous world of agricultural chemistry. We're going to be walking through the complete experimental details presented by Dr. Chem on the determination of phosphate in fertilizers. If you've ever found yourself wondering how we actually know what's inside the fertilizers that literally feed our planet, well, you are in exactly the right place. We are going to explore the precise, step-by-step procedures a chemist follows to figure out the exact amount of available phosphate in solid fertilizers. It's a process that requires some incredible precision, some really fascinating chemical reactions, and a little bit of clever math. So let's just dive right into this.
To really grasp why this analysis is just absolutely critical, we kind of have to start with the biology. Phosphorus is a primary macronutrient. Without it, plants simply cannot create DNA or ATP. And ATP is basically the energy currency of the cell. But here's the catch. Plants can't just absorb raw phosphorus out of nowhere. They need it in a highly specific, accessible form, primarily as dihydrogen phosphate. So when a farmer goes out and buys fertilizer, they're looking for the exact guaranteed percentage of available phosphorus pentoxide, or P2O5. If that label on the bag is wrong, well, crop yields suffer. So accurate laboratory measurement isn't just some annoying regulatory hoop to jump through. It is the absolute bedrock of agricultural success.
All right, so our roadmap for today looks like this. First, safety and apparatus. Second, the gravimetric procedure. Third, a fun little section called the silica villain. Fourth, the final calculation. And finally, number 5, modern rapid methods.
Okay, let's kick things off with Section 1, Safety and Apparatus. Actually, before we even think about touching a sample, Dr. Chem is super clear on this. Safety is paramount. We are dealing with some seriously intense reagents here. Nitric acid, it's highly corrosive to both your skin and your equipment. And aqueous ammonium hydroxide produces these toxic, really nasty vapors that can severely irritate your respiratory tract. Because of that, preparing these chemicals requires the mandatory use of fume hoods, specialized eye protection, and heavy-duty gloves. So this isn't your average baking soda volcano high school science experiment. This is rigorous, high-stakes, real-world chemistry. To pull off this specific extraction, you're going to need some highly specialized gear. We rely heavily on an ultra-precise analytical balance. We're talking weighing samples down to the 10,000th of a gram. You'll also need a water bath equipped with a shaker that can hold a perfectly steady 65 degrees Celsius, some heavy-duty 250 milliliter boiling flasks, a 250-degree laboratory oven, and, maybe most importantly for this whole thing, Gooch crucibles, specifically Coors No. 4 crucibles fitted with glass fiber filter paper for the final vacuum filtration step.
Moving right along to Section 2, the gravimetric procedure.
So the whole process kicks off with a raw half-gram sample of solid fertilizer. We add exactly 100 milliliters of an ammonium citrate and EDTA solution, and then secure that boiling flask into our 65-degree water bath to just shake away for about an hour. Now, what's really interesting about this step is the specific role of that EDTA. You see, fertilizer is packed with all sorts of various minerals. The EDTA actually acts as a complexing agent specifically targeting calcium and magnesium. It basically binds them up so they don't accidentally precipitate later on and ruin our careful phosphate analysis. It's essentially a brilliant little bit of chemical crowd control. After that hour of shaking is up, the sample gets diluted with water and is allowed to settle. Then, the chemist has to pipette a very specific aliquot, which is just a fancy word for a precisely measured portion of the clear liquid. But here's the cool part. The volume extracted depends entirely on the guaranteed P2O5 percentage printed right on the fertilizer's ladle. If it's a super high-yield fertilizer with over 19% phosphate, we only need to pipette 15 milliliters. Why? Because this entire protocol is meticulously calibrated to form exactly 0.3 grams of precipitate in our very next step. Like I said before, precision here is literally everything.
Okay, moving right into the precipitation phase. We add 10 milliliters of nitric acid and bring the whole solution to a boil. Then, we introduce 50 milliliters of Quimiciac reagent. And guys, this is where the magic really happens. The Quimiciac reagent reacts directly with the available phosphorus, pulling it right out of the liquid solution and forming this solid, bright yellow precipitate known as quinoline phosphomolybdate. We boil that briefly for about a minute, let it cool down, and then use our vacuum filter flask to forcefully pull the liquid right through our Gooch crucible. This leaves only that solid yellow precipitate trapped on the filter. Finally, we bake that crucible in the oven at 250 degrees Celsius to completely remove every single drop of moisture before weighing it.
Which brings us to Section 3, the silica villain.
So sometimes our fertilizer samples, particularly those pulled from environmental sources, can contain pretty high levels of silica, and silica is an absolute master of disguise in the laboratory. It can actually react with our reagents to form a silicomolybdate complex. The problem? This silica complex perfectly mimics the phosphate complex we are actively trying to measure. To the naked eye, and crucially, to our highly sensitive analytical balances, the extra weight from the silica looks exactly like the phosphate. This causes unexpectedly high false readings that can completely trick the analyst and totally compromise the integrity of the data.
So you might be wondering, how do we defeat this interference? Enter the hero of our assay, oxalic acid. When troubleshooting these unexpectedly high or variable results, chemists will smartly adapt the protocol by adding a 0.25% oxalic acid solution to the mix. What it does is amazing. Oxalic acid selectively attacks and destroys that fake silicomolybdate complex within about 5 minutes, but, and this is the key, it leaves our true target phosphomolybdate perfectly intact. It essentially cleans up the sample so we can actually trust our numbers again. Pretty neat, right?
All right, on to section 4, the final calculation.
0.0327. Dr. Cam highlights this highly specific number as the gravimetric factor, and it is pretty much the linchpin of our final result. But where in the world does it come from? Well, it's derived mathematically directly from molecular weights. You take the molecular weight of the P2O5 we are looking for, which is 141.90, and you divide it by twice the molecular weight of our massive, bulky quinoline phosphomolybdate precipitate, which sits at 4,425.84. Do the math, and the result is this constant factor. It represents the exact mass ratio of the target molecule to the total precipitate molecule.
And the final equation just brilliantly illustrates how everything comes together at the end. We take the exact oven-dried weight of our precipitate, multiply it by our dilution factor, multiply that by our gravimetric constant of 0.03207, and then multiply by 100 to convert it to a percentage. Then we take that whole top line and divide it by the original raw sample weight. This beautiful bit of math is exactly what determines the final reported percentage of available phosphoric acid. It's incredibly elegant, but as you can probably tell from all these steps, this traditional method takes a very long time, often spanning overnight.
Which perfectly sets up section 5, modern rapid methods.
So research published in the Pakistan Journal of Biotechnology focused exactly on this time bottleneck. In the traditional standard method we just discussed, that yellow precipitate is left to settle overnight. That means the full analysis literally takes 2 days. However, researchers have successfully validated a modified rapid method where the sample simply settles for 3 hours inside a standard refrigerator. The results? It's a massive, game-changing time saver while still maintaining an incredible 98 to 102% accuracy recovery rate. It makes it perfectly suitable for quickly advising consumers without that frustrating 2-day delay.
But some modern labs are pushing the envelope even further. We're now seeing the rapid rise of the thermometric titration alternative. Instead of meticulously drying and weighing a precipitate, this method determines phosphate via a precipitation titration with magnesium. The equipment literally tracks minute temperature changes as the chemical reaction occurs to find the endpoint. It requires absolute zero sample preparation for liquid fertilizers. And the best part? Instead of taking hours or days, an entire determination takes about 5 minutes. 5 minutes! That is a real game changer.
So we'll end things today with this thought. As global agricultural demands continue to skyrocket, testing laboratories are under immense constant pressure to deliver faster results. Will the slow, historic precision of gravimetric analysis, with its heavy ovens, specialized crucibles, and overnight settling, eventually be entirely replaced by these rapid 5-minute thermometric titrations? It's a truly fascinating balance between traditional rigor and modern efficiency, sparked by Dr. Kem's incredibly detailed look into agricultural chemistry. Thank you so much for joining me on this explainer. I hope this sparked your curiosity, and I will catch you in the next one.